I have to leave early today. I know you're upset, but I had to schedule a doctor's appointment. That's pretty important, and the only time they get me in was noon. So we're just going to go till 11 today, because that's kind of factor in travel time. Which this is not the greatest day to have only 3 hours to deal with stuff. What I'd like to do to try to get as much stuff covered as possible is to not take a break. Or if you'd rather think of it this way, we're going to take a break at 11 o'clock, and you'll have a whole week off before you come back. So, just in the interest of getting stuff done. So if it's coming up on 9:45, and you're like, oh, I can wait 5 more minutes till we take a break. We're not taking a break. So go ahead and, if you have to go to the restroom or something like that. You're adults, you know when you need to go. So, okay, before we get underway, I do want to talk a little bit about last week's test. I still don't know why the clickers didn't work. Because my AP1 class took theirs yesterday, and the clickers worked for them. So it was just sort of a weird little fluke. And all I can guess is that weird little fluke was some higher power forcing me to grade the test by hand. And in doing that, I got to see a little bit better how people were answering questions. Normally what I do in the program that the clickers work with, it tabulates all of the scores, and I can see how people do, and I look across for which questions people did poorest on, that sort of thing. I actually don't have the data for that, the statistics. I have to by hand put in everybody's answer, and I don't want to do that. So I just sort of went with my impression as I was going through. It did suggest to me something that's not going to help you a whole lot on that test because it's passed, but hopefully I'll be able to get something together before the second practical. I think I need to do better with some sort of study guide. And I think I need to do better with some sort of study guide because I think people are not paying attention to some really fundamental aspects of this course. And to illustrate that, I want to pull up these 2 questions here, which you might recognize. Oh, sorry, those aren't the ones since I changed the magnification. Where'd it go? Okay, so these aren't the ones I meant to pull up, but the point I want to make is actually kind of similar. I'll go to the other ones that I wanted to. And it's really just this question 6 here that I want to make a point about here. And then there's a pair of questions that the same point kind of applies to. So to remind you, the station for question 6 there was that model up there on the board. And you had to identify what the pointer was on. And then the question was asking what neurotransmitter would be released by that neuron. Now, for the autonomic system, there's only 2 possible answers for any question about neurotransmitters. What are they?
Acetylcholine.
Yeah. So acetylcholine is used at 3 of the 4 synapses in the autonomic system, and norepinephrine is used at the other one. I wrote the answer choice there, nor, in parentheses because just to be as inclusive, but really it is norepinephrine and not epinephrine. So glutamate and serotonin cannot possibly be the answers there. Those are just not neurotransmitters used in the autonomic system. And I had a number of people putting B or C for this answer. I can't remember offhand which one it was, but there's another question along the same lines, or 2 questions. Where are they? Oh, I know. I did have them up on, nope. Oh, it's in the other case. That's right. Okay, so these two questions, 20 and 21 here, again about neurotransmitters. In the case here, it's about peripheral neuropathy, which can affect postganglionic neurons in the autonomic system. And it's again asking about neurotransmitters. So the two questions are asking about which causes dilation, which causes constriction. And there's only two neurotransmitters that are reasonable answers. Again, it's going to be norepinephrine or acetylcholine. Okay. Now, the fact that these are opposing processes, the pupil doing one thing versus the other, it should be evident that one answer is one of those two things and the other answer is the opposite. That's the way the system works. Any given, hold on a sec. Where is that? There we go. Any given target organ is going to have opposing processes, and the only way it knows what to do is depending on which of those two it gets. Okay. So I had some people that would answer, say, 20 correctly, and then for 21 would put B or C. I had some people that got both of them wrong because they put A for one and D for the other, but got them backwards. So that's an acceptable wrong answer. If at least your answer is only norepinephrine or acetylcholine, you're in the right neighborhood. And if you get them backwards, I understand why that might happen. But answering glutamate or serotonin just doesn't fit with the way that this should work. So, so I just wanted to real quick. Oh, stupid thing. I just wanted to real quick point those kind of questions out as an example of the kind of things that I have to assume fundamentally no. Okay, that's a basic point about the autonomic system. I wrote the chapter on the autonomic system. I know I made that point in there. There's a table that shows the four different synapses, and by that I mean the preganglionic sympathetic, the preganglionic parasympathetic, the postganglionic sympathetic, and the postganglionic parasympathetic. And in that table, three of the things in there are acetylcholine, and one is norepinephrine. Okay. So there are a number of things that were kind of at that fundamental level, some things that I should expect that A&P students, A&P2 students should know in the course. Some of them are things that should have been addressed in A&P1, again, fundamentals, but they might not have been covered this semester. They have to be able to assume that you retained information from A&P1. So I'm thinking I need to put together a study guide sort of thing a little bit more to remind people about these fundamentals. And I think I haven't quite figured out how I want to do this, but I think I need to not spoon feed you the information, like put a point on a piece of paper saying, remember, the autonomic system only has two neurotransmitters, but rather something like a question that gets you to look up what are the neurotransmitters in that system. Or I'm thinking about putting something together that's more like an assignment, not a graded assignment, but a worksheet. And the thing with worksheets is that you want the feedback on, if you put the correct answers in. So I'll have to set something up, but I could do like a matching with all sorts of different neurotransmitters, for instance, and you have to pick which ones are somatic, which ones are automatic, autonomic, which ones are both, something like that. Again, I'm not quite sure how I'm going to do this, but I think that might be something I need to do as a preparation, which actually would be kind of nice because I'd like to replace the outline thing that I have. I just don't like those outlines. They're old, they might be a little out of date, they might not be organized quite so well or something like that, but maybe a study guide would be a better approach there. So just in going through and seeing what people actually answered, because I looked at everybody's answers rather than having the computer do it, I saw a lot of things where people seem to be answering questions without thinking about the fundamental information that the question's about. So that's a component of the questions that I ask, and it's, whether it's in a Blackboard assignment or in those kinds of tests, and you need to think about what the question's actually getting at. What's the point of the question? That's sometimes integral to knowing what I'm looking for an answer. So there you go. Now, we certainly don't have time to go over the test. I've never had time to do that. I've never taken time out of the class to sit and go over the test with the whole class, and I don't think that's necessarily helpful. Some people would appreciate it, but the majority of people would just sit there and check Facebook or whatever you do. So I don't do that, but if you're interested in looking over your tests, you're always welcome to do that. I keep the paper tests for a year after the class. If you go into my room, you'll see that there are some piles of paper, and that's what they are. They're the practicals there. Blackboard stuff is only there until grading takes place, because after I finish grading for a semester, I have to archive the course to save all the data if anything comes up. The process of getting that archive data back is prohibitive and only called upon in extreme cases, which is practically never. But it's out there to be kept on hand if something comes up. But the paper tests I keep in my office. You're always welcome to come by and look at them. I'll give you the answer sheet. You can see which ones you got right and wrong. Actually, since I hand graded these, they have check marks on them. So you don't need to see the answer sheet. It shows you which ones you got right, and then we can talk about the ones that you got wrong. So so you're welcome to do that. Yes.
Would that be during office hours?
Well, yeah, I have to be in my office for you to be able to come by and look at your if you'd like to contact me and say you want to take a look at it, and right after class isn't good for you, like today, it's not good for me right after class. But if next week you want to look at it right after class, what am I doing next week? Oh. No, I have a meeting right after class next week. Anyways, if you'd like to look at it and you want to by email, we'll find a mutually agreeable time, Tuesday afternoons after my MP1 class, my workout or something like that. But yeah, I do have to be present and we have to get into my office. And so we'll find a mutually agreeable time to do that. So anyways, so that's all I want to say about the test here in class. And hopefully I'll have some better idea of how I want to approach this before the second practical, but we'll see. Now, like I said, I have to leave early, so we only have until 11 o'clock. And I don't want to take a break so that we can make the most of that time. And this is basically an outline of the information that's important in this chapter. So this is essentially what I want to do. I'm going to kind of move through things rapidly. That's kind of my approach. Feel free to stop me and ask questions. I'm in this mode of, I gotta get this done by 11 o'clock, so I'm going to try to move through things quickly. But more important than covering everything is being sure that you can understand as much as possible. So as I'm going through this, please feel free to stop me and ask questions, and we can deal with confusion as it sets in as best we can. So as I was and I kind of wish I'd known that this was going to be a short day last week, because I felt like what I did with the time we had after the test yesterday now is kind of a waste of time. Now, when I was playing the podcast, I was writing stuff down and up on the board, so okay. So I'm not going to go back and talk about that. I will talk about, from that podcast, there was mention of attack cells and lullaby cells, and I'll point those out and what parts of the immune system that's referring to. So you can kind of fit it into this. But I'm not going to talk about it. An important idea in the podcast was the hygiene hypothesis that one of the speakers in there brought up, and I'll try to fit that in with some of what's going on here, but we'll see. So one of the things I did last time was I kind of set up what the immune system was. I talked about innate and adaptive immunity as the two components. And then within that, I spent some time talking about active and passive and naturally acquired and artificially induced, I think are the terms that I used, just to characterize at least the adaptive immunity stuff. And from that, it might seem like I spent more time concentrating on the adaptive immunity, like that's the big thing, and innate's not that important. That's not really true. It's just innate immunity is not as involved or complex, I suppose. So a little bit more time spent talking about adaptive immunity. Innate means to be born with, and so these are the things that are in place at the time of birth. Another way of thinking about innate immunity is that it's not specific or non-specific. These are ways that you have to deal with any threat to your system. And by that I mean infection, injury, those kinds of things. The immune system is responding to more than just pathogens, although that's what we kind of concentrate on here. And conversely to that, the adaptive immunity is specific, which is how we deal with a particular pathogen, like the influenza virus, without worrying about other things. When that particular pathogen is causing problems from your body, the adaptive immune system is there to do its job. So for textbook presentations, that's innate, adaptive, non-specific, specific. Usually a way to think about these, but really it's all one big immune response. And the innate immunity is sort of the aspects of the response that are quickest and sometimes are all we need. If these don't do their job, then the adaptive immune response aspect of it will come online. They're all going to start at essentially the same time. They're all activated by similar signals. It's just the adaptive response takes a little longer to do its job because it's targeting the specific thing. The adaptive immune response will make use of some parts of what I'm about to describe as innate immunity because it needs ground troops, if you will, to do the work. And some of the non-specific things directed by the specifically targeting adaptive response can be used in an adaptive way as a tool. So they get all mixed together, which is really the entirety of the immune response, even though we kind of break it down this way. So with innate immunity, I want to talk about these various things. The book goes through them also, not exactly in the same way or order, but this is the way that I want to kind of address it. So going to the book, let me just use some of the figures here. Barriers are talking about just physical limits separating us from the outside world. Now, usually when we think what is us versus what's the outside world, we think of what's within versus beyond our skin. That's our usual cognitive perception of ourself. Immunologically, that's not exactly it. The skin is a very important barrier. However, the mucosal lining of your digestive tract, your respiratory tract, your urinary tract, and your reproductive tract are also barriers. So whether something's outside of you in the kind of cognitive sense or in your digestive tract, but not across the digestive mucosa, immunologically, they mean the same thing. You can swallow anything you want. I don't recommend it, but your system can handle that you'd swallow anything you want as long as it doesn't cross that barrier. Now, it's not a magical barrier. It's not that you could swallow anything you want. There are a lot of things that get across that barrier. So swallowing broken glass is definitely going to shred up your digestive mucosa. It's going to cause a lot of problems. You can swallow certain pathogens and they can't get across the barrier of your digestive mucosa, and your stomach acid and all that destroys it. But some things can survive that or get across the digestive mucosa and cause problems before they get to the stomach. And I use the digestive system as the primary example just because I like to throw in the thing about the stomach as being largely antimicrobial, which is important. But you can think about the same thing for the respiratory system or the reproductive system or the urinary system. This picture in the book is kind of addressing some of the things that play a role in those barriers. And the picture is largely concentrating on the respiratory tract. And it's just showing that there are components to, in this case, respiratory mucosa that helps protect us against pathogens that we might breathe in. The kind of things that you'd find, which applies not just to this mucosa, but to the other barriers, the other mucosal barriers and the skin, for that matter, are certain chemical components that are secreted. On your skin, a lot of secretions have a little bit of an acidic pH. Whatever pathogens are trying to get into our body are trying to live in an environment that's about 7.35 pH, which is our physiological pH. The outside of our skin is a little bit acidic relative to that, which can inhibit bacterial growth of the types of things that are trying to get into our body. Or things that like living in a slightly acidic pH, and by slightly acidic, I mean about 6.5, they wouldn't be comfortable in the pH inside our body. So it helps to create this kind of barrier between the two. There are other things that are secreted from the skin, not so much, but in mucosal linings more so. Specific chemicals that are antimicrobial. Enzymes that break things down, primarily lysozyme. Lysis means to break down, and so lysozyme is an enzyme that breaks pathogens down. And it's found in mucus secretions in various places in your body. There are cells that play a role, white blood cells, which we'll talk about, and some of them are pointed out here. The book uses this cell right here as a common example of other functions that we're going to talk about later on. And this is what's called a dendritic cell. Now, the word dendritic means branched. So it's a cell that, looking at it in a microscope, has a bunch of branches coming off of it. That's where the name comes from. But in the skin and these mucosal barriers, this type of cell is prominent, or I shouldn't say prominent, but present, to help alert the rest of the immune system when something crosses over. So if you get a cut in your skin, and bacteria can cross over and the skin stops being an effective physical barrier, dendritic cells in your skin will then recognize the presence of a pathogenic invader and be able to alert the rest of the immune system to help fight it off. Dendritic cells are actually not that common compared to some of the other cells here, and the book uses it quite a bit, which might seem like they're fairly common cells. But the reason the book's using that is somewhat to set itself apart from other books. So I'm going to talk about some functions of certain cells, and the book will have a picture of a dendritic cell as an example, but it will actually apply to other cells just the same. Now, if you think back to the skin chapter, dendritic cells were mentioned. I specifically use that name, but they have a more particular name for the skin. In the stratum spinosum, a living layer of the epidermis, there's a type of cell called the cell of Langerhans, or Langerhans cell. That's just the dead white guy name for the dendritic cell in the skin. So I use dendritic cell when I'm talking about that, because it sets up an understanding that that's a component of the immune system. You will not see, other than in a parenthetical remark in this chapter, reference to the cells of Langerhans. It's going to be talking about them as dendritic cells, which is a more appropriate name for them. And that's what's pointed out here. Down below, they have representations of B cells, B lymphocytes, and T lymphocytes up here, which are also going to play a role in immune responses. They might be present at the barriers to be ready to fight off infections when they come in, but there's a lot more to how lymphocytes work, and we'll talk about them towards the end of the day. This is what the book has to represent the idea of barriers, but keep in mind that it's more than just the nasal mucosa. The other mucosal linings and the skin all play a part there. And of course, you're born with those, so it fits into the innate system. Here's a little table pointing out some of the things that are present in the skin, the digestive mucosa, and other mucosal surfaces that play a role in helping defend the body as barriers. At the bottom of this, kind of interesting, until this book, I'd never really thought of mentioning it here, but the last entry here says normal flora, or non-pathogenic bacteria. We have a lot of bacteria that live in our body. I have seen sources say, and I've not verified this, and I don't really have any reason to doubt it, but there are more non-human cells in you than human cells. Okay? There's a lot of bacteria that live in and on your body. And our body's a great environment for them to live, and they want that environment to persist as long as possible. So they seem to play a part in helping immune function. They want to keep the environment that they live in healthy, and so they can play a part, especially those that are on the mucosal linings, especially in the digestive system. There are non-pathogenic bacteria that lend their support to keeping us healthy, and that's what's mentioned here. Now, just to jump to the little outline I put here, I have written here phagocytes, just as sort of the premier example of what I want to talk about here. But really, what I want to cover for a second are the different types of cells that play a role in the innate system. And phagocytes are the main one. There are two types of cells that are phagocytes, macrophages and neutrophils. Now, there are 5 types of white blood cells. 3 of them are called granular leukocytes, because of how they appear underneath the microscope. Neutrophils, eosinophils, and basophils. Okay, all of the granular leukocytes have "phil" at the end of their name, which is describing how they react to stains when we're looking at them under the microscope. Then there are two agranular leukocytes that don't have grainy looking appearance under the microscope. There are monocytes and lymphocytes. Now, lymphocytes are different from the other types of white blood cells, and we're going to deal with them separately because they're part of the innate system, the adaptive system. The other types of white blood cells essentially all fit into the innate system, but the relative numbers of these different types of cells means that we're only going to deal with thinking about two of them. Now, really, if you look at the different types of white blood cells, all five, the way to think about them as in regards to their relative number in your body is neutrophils are the most common. They make up about 2/3 of the white blood cells in your system. And that's followed by lymphocytes, which are about 20 to 25% of the white blood cells in your system. And then monocytes, which are a little less than 10% of the cells there. Now, if you think 2/3, 20 to 25%, and a little less than 10%, you can make those three add up to 100%. Okay. Now, the numbers are a little rough, just to give a picture there. And so there's still room. We haven't completely populate our body. So the other two, the eosinophils and the basophils, and in that particular order, are the next most. But they collectively make up a very small fraction. It might be a couple of percent total or something like that. They're pretty rare cells. One way to remember the 5 types of white blood cells, and especially in their decreasing a number of population, whatever, is the mnemonic sentence, "Never let monkeys eat bananas." Okay. Never for neutrophil, let for lymphocyte, monkeys for monocyte, eat for eosinophil, and bananas for basophils. That in that order are the most to least numerous white blood cells. And I just gave you a rundown of their basic number. About 2/3 are neutrophils, 1/5 to 1/4 are lymphocytes, and less than 10% are monocytes, and then a very, very small fraction of the other two. Now, lymphocytes are different. Like I said, we're going to talk about them with the adaptive system because that's what they're about. So that just leaves for us the neutrophils and the monocytes. Now, if you've been looking at this, what's on the screen here, you don't see in the big header there anything that says monocyte. It says macrophage and neutrophil. Macrophages are monocytes. Monocytes are what we call them when they're sort of everyday cells, part of the blood system. When they get activated and they become an important part of the immune response, they're macrophages. And if you look at what the book has here, there's actually 3 little paragraphs, each starting off with a bold-faced name. So macrophage, neutrophil, and monocyte. And what it says about monocytes here, which is really just a sentence tacked on the end, is that they are what become macrophages. Now, phagocyte means eating cell. Phago, as in the root for esophagus, is referring to eating. And cyte, it means cell. Now, exactly how you interpret that, either these are cells that eat or these are things that eat cells. Either one's essentially appropriate. Although I usually think of them as cells that eat because they eat more than just cells. They are highly mobile cells. They move around like amoeba, and I showed you an amoeba last time. I'm going to show you a couple of videos that illustrates how these move, because it's kind of interesting to watch. What they do is they find things that aren't supposed to be in our body. That doesn't have to be pathogens, but that's what we usually think of. But they're also important for cleaning problems out of our body, whether it's debris of tissue that's been damaged, old red blood cells that have lived their lifespan, diseased cells in our body. And one of the videos I'm going to show you is actually of cancer cells, and those sorts of things. So they're kind of like the vacuum cleaners of our system. Okay, so I want to show you a couple of videos. In the video content section for the course, I've laid out a bunch of videos that have to do with all the stuff that we go over. And just when I come across something that I want to use in the class, I'll usually put them in here. And they're in order for how we go through the course. So it starts off with autonomic system, endocrine system, cardiovascular system. Come on. And then after the cardiovascular system, it gets into the immune system. So this is not one I want to show you, but if you're interested, I just put it in there. It's kind of interesting. It's A through Z, and the words they use for each letter are something that have to do with epidemiology, which is related to immunology. This is the first video I want to show you. And this is very brief. There's no audio component to it. I kind of feel like it needs a little player piano in the background. And what we have here, Liz, could you kill the lights real quick? There you go. What we have here is a phagocytic cell. It's probably a neutrophil. That's what the title up here says. And it's chasing down some sort of bacteria. The cells around in the background, these are red blood cells. So you can see sort of get a sense of scale here. And we're going to see this cell moving and chasing this thing down and finally engulfing it and pulling it in to be digested. So as the neutrophil is moving along, the leading edge of the cell is kind of pushing out, and it's actually pushing the red blood cells away as that happens. And eventually, it pulls the bacterium inside of itself, and we don't see it in this picture, but in this video, but that bacterium is going to be digested and broken down. And that's how phagocytes work, basically. They chase things down in our body and get rid of them. Yeah.
Is that real time or is it sped up?
I have no idea. I never really thought about that. And the reason why I don't want to take a guess is because the other video I want to show you, which I found this past semester, and I haven't put it in the summer course because I wasn't thinking along those lines. So let me move over to another place, and I'll show you how to get to this video when it comes up. Shoot, that's AMP1. Okay, so actually the link I have here doesn't go where I really want to go. Where is it? There it is. So the way that what I have here in Blackboard works is I can go to a special place in Blackboard and I can find YouTube videos and they get embedded. The thing is that YouTube videos that I can embed have to be publicly available on YouTube. And so this video here is made by the same people as the one I'm about to show you, but it's their publicly available one. I did put down here a link to a website called LabRoots, and since this video, what I have in front of you on the screen right here, actually isn't in the Blackboard site that we're using, let me just tell you to look for lab roots. And as I'm holding the mouse over the link down there, if you pull this up in the recording, you can see down in the bottom the full URL if you want to go directly to it. Or you can see it up here at the top of the screen. This is a YouTube video, but for some reason they didn't make it public on YouTube, and it's just embedded in this particular page here. Now I want to play this because it does show you what neutrophils look like, and it's a great video showing how they move. But it's going to address a couple of things that are important with adaptive immunity, or that we don't want to deal with just yet, as far as the course is concerned. I mean, as far as my presentation is concerned. So let me play this out, and at the end I'll mention a couple of things, and I'll say, remember that, we'll talk about that in a couple of hours. So, and there's a voiceover here. After a lot of introduction things. I don't know what this picture is about. Oh.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer...
Okay, actually I'm going to stop there. For the sake of what we're doing right now, that little sequence is what I wanted to show you. I had said I was going to show you neutrophil, and I just sort of blanked on what this was actually showing us. This is actually showing us a type of T cell, which is something we're going to talk about later on. But as far as this video, this little sequence here, starting at about 47 seconds, here's one of these cells, and it's a great representation of how these cells move around. Now you can see a kind of shadow of some cells in the background. But what they've done is they've stained a particular protein in this cell with a fluorescent marker. And they're shining a UV light on this, causing that fluorescent marker to light up so that we can see it in the microscope. You don't see the fact that it's moving through an environment with a bunch of other cells. It's probably in a petri dish, and these are just some tissue cells from wherever they're doing the research. But they've just highlighted the T cell so we can see how it moves. And you can see how its cell membrane pushes out these pseudopods, and those sort of pull it forward. Okay. And even this little sequence right here, come on, back up. I think that was it. It's sort of moving over a cell. There we go. So it's crawling over top of this cell, and we can see that cell, you know, as a very, very light background image. I don't know the exact methodology they use here, if it's something about the stain that they're using, that it highlights cells that touch the cells that are crawling, or what, but we can see a little bit of that there. So this is a good representation of the idea of how these things move. Now I'm going to come back to this video because the next thing it gets into, what I stopped it at right here, now we can see it's showing our the cell we were just looking at, not the exact same cell, but same kind of cell. And then in blue is a cell that it's going to destroy. And this is about cancer, so this is a cancerous cell. And so when we're talking about adaptive immunity, and these are the cells going through the adaptive response. I'll come back to this and finish this video, but that first sequence there is a great representation of how the cell moves around. Now, I put phagocyte in the little outline of what I want to do today, but really it's what I wanted to talk about were the types of cells that are part of the innate response. And there's one other type of cell, which is called a natural killer cell, or sometimes just referred to as an NK cell. NK cells are lymphocytes, but they're non-specific lymphocytes. So they're not T cells, they're not B cells, they're their own kind of lymphocytes. And what they do is they destroy other cells. So actually the part of the video that we're going to get back to and look at, that T cell is killing a cancerous cell, natural killer cells do that also, but they do it in a more non-specific way. Anything that doesn't look like it belongs in your body will be destroyed. Okay. Now, this next little thing here that says recognition of pathogens addresses what I just said. Anything that looks like it doesn't belong in your body. These cells, whether they be phagocytes or natural killer cells, the way they do their job is they look for the presence of things that don't match what's supposed to be in your body. You have cell surface antigens, proteins on the surface of your cells, that basically let your immune system know that the cells in your body are supposed to be cells in your body. Anything that doesn't match that sticks out like a sore thumb, and these cells, phagocytes or NK cells, will destroy them. In this little section that says recognition of pathogens, it has in boldface pattern recognition receptor. Every cell in your body is going to have a slightly different cell surface signal to say that it's supposed to be in your body. Like red blood cells have the blood typing antigens that we talked about with that topic. But different cells have different things. But what's consistent is the type of molecular pattern that they represent. So these cells have these pattern recognition receptors that can recognize things that are supposed to be in your body. And when they come across something that's not supposed to be in your body, they can destroy it. Bacterial pathogens especially are a great example of how this works. While bacteria often have components to them that make it possible for them to move, like cilia or flagella or something like that, a lot of them don't have any active movement components because they live in liquid environments and they just let whatever liquid that they happen to be living in move them around. And more importantly is how they stop moving. Bacteria will be covered in glycoproteins, which would be carbohydrate structures attached to proteins that are in their membrane, or glycolipids, which would be carbohydrates that are attached directly to the lipids in their cell membranes. The carbohydrate part of both of those, the glyco part of both of those terms, makes them sticky. Just like if you were covered in carbohydrates, i.e. sugar, you'd be sticky. And it just is a matter of how carbohydrates interact with other hydrophilic compounds. And they're going to be attracted and form a bunch of hydrogen bonds. And so being covered with carbohydrates, they're sticky and they can stop and stay in an environment that they want to stay in because they stick to it. The thing is the stickiness of their cell membrane is also unusual. The cells in our body stay in place through different means, so they're going to look different. So that kind of pathogen is going to be recognized as not part of your body because it contains that, and it's going to be destroyed by one of these cells. Now, basically, the phagocytes destroy things smaller than cells in our body. Okay? We just saw a neutrophil hiding down a bacterium moving around an environment filled with red blood cells. Okay? So the size there gives you an impression of what they kind of go after. Now, there are macrophages that are specifically for getting rid of red blood cells, and red blood cells are small cells. But as far as pathogens are concerned, it's usually dealing with small things. NK cells deal with cells in our body that have problems. So if it's a cell that's been infected with a virus or cancer cells or something like that, that doesn't look right, NK cells will destroy them. And in the text here, the bold-faced words under natural killer cells, like perforin and granzyme, those are chemicals that they use to do that. And actually, the video that I stopped, when we get back to that, those are the chemicals that are used to destroy them. So we'll see how that actually works in a video. And that's really kind of the most impressive part of how that video is put together. Now, the next thing that I want to look at, I just put, well, I put a couple of things. Complement and interferon. These both belong to what the book is calling soluble mediators of innate immunity, or the innate immune response. There are a number of chemicals, mostly proteins, that play a role in coordinating the immune response, and they're part of the innate immunity. So I'm going to use the book here just to mention a few of them, but I want to concentrate on complement and interferon. And actually, now that I realize it, the book puts interferon before complement. So I'll talk about that first, even though that's not the order I added in my outline. So I'm not going to talk about these a whole lot, but cytokines and chemokines, those are classes of hormones. So those are how white blood cells communicate with each other, and sometimes with other cells that have to do with immune function. So those are really just hormones, and we've talked about hormones. Then the next thing that's mentioned here are early induced proteins. And one type of early induced protein, one class of early induced proteins, is called interferon. There are actually a few different types of interferons, and I only want to talk about one type because it's important for how we deal with viruses. Now, an early induced protein is just a protein that cells make very quickly after they get signals about the presence of pathogens or something like that. Okay. It's cells, when they're doing their daily work, will activate genes and make proteins and that sort of thing. Early induced proteins are just the earliest of those that are a component of immune responses. So interferons fall into this category. There are alpha interferons, beta interferons, gamma interferons. I think it's just those three. And I can never quite remember which one it is, so I'm not going to say. But one of those, alpha, beta, or gamma, is an early induced protein related to viral infections. Now, the other ones are made by white blood cells. So when white blood cells are activated, they make their types of interferons that have a role to play in the immune response. But other cells in your body, when they're infected with foreign DNA or foreign genetic material, they will immediately start making a protein, early induced protein called interferon. And again, I can't remember which one it is, so I'm not going to say specifically, but it's one of the types of interferon. And it is a hormone or signaling molecule that's released from that infected cell to let all of the neighboring cells know that viruses are in the neighborhood. Okay. So it's sort of like a burglar alarm. So if somebody tries to break into your house and they set off the alarm, that alarm lets everybody else in the neighborhood know that there are burglars in the neighborhood. However, interferon is like a silent alarm. Okay? So if somebody tries to break into your house, part of the point of having an alarm is to scare them off so they don't steal anything. But with interferon, the alarm goes off to let all the neighbors know that there are viruses there, but the virus is not going to respond to that also. The virus is going to infect that cell. So that's sort of like if you had an alarm that let burglars break in and they could steal whatever they wanted to, but it also alerted the neighborhood and the cops or whatever. And there'd be a response to deal with the burglars eventually, but you didn't care what happened to your house. That's kind of what interferon's doing in viral infections. The reason for that is that when viruses get in, they inject their genetic material. That genetic material is going to hijack the cell, and it can't undo that. It's going to force the cell to copy that genetic material to make more viruses and to make viral proteins to package up that genetic material to be sent on. And that's usually going to happen at the expense of the host cell. The host cell will probably die making copies of the DNA of the virus and until it bursts open or something like that. And at the point when a virus has infected a cell, there's not a whole lot the cell can do. It's got to, take one for the team, so to speak. But what it does is it lets all of the neighboring cells know that there are viruses in the neighborhood, and it'll keep them from being exploited in the same way. However, the way it does that is rather extreme. Interferon, when it binds to the interferon receptor on a cell that has not been infected by a virus yet, will cause protein synthesis to stop in the cell. It'll just block the production of any kind of protein. Now, cells need to make proteins all the time for their normal metabolic processes. If interferon shuts that down, then the cell is like doing nothing. It'd be like if you held your breath until the burglars got out of your neighborhood. Okay. There's a slight chance that the cell can survive that if the viruses are controlled and the cell can go back to being active. It's possible, but more likely that all of the cells are going to die. It's just the initial one was infected, it's making more viruses, and all the rest, which are going to die, didn't get infected with viruses, and so the virus won't be able to spread. Usually viruses are infecting epithelial tissues, so an influenza virus is going to be airborne, it's going to get into your respiratory tract, and it's going to infect the epithelial cells there. All of the symptomology that you get from the flu is really about how that epithelium is affected, increased mucus production, that kind of stuff. And you ride that out until the epithelium can repair itself. And epithelia repair themselves every day. They're very good at it. So all of those cells that die to limit the spread of the virus are doing it because they know that they're going to be replaced by new cells and you'll be fine. Not that cells actually know that they're giving themselves up for the greater good, but it's essentially what's happening. In other situations, viruses that infect other types of cells, it can create bigger problems. For example, HIV infects a type of lymphocyte, the T cell, and so when it's active and it's killing those cells off, you lose white blood cells and you lose immunity, immune capability, which is where AIDS comes in. It's acquired immunodeficiency syndrome, AIDS, because T cells are being destroyed, and T cells cannot be as easily replaced as epithelial cells. Whether they're actually being infected by the virus or something like interferon is protecting them. If they die, and you lose that immunity. So that's basically what interferon is. There's not a picture to depict that. So hopefully my analogies to the burglar in the neighborhood and that sort of thing illustrates it kind of better. But, then I want to talk about complement, which technically I put in the list before interferon, but the book does it second, so we'll deal with it in this order. Now, the word complement is a homonym for the word complement. Okay? And that's what homonym means. Two words that sound exactly the same, but are not spelled the same and do not mean the same thing. So complement in this sense means to work together, sort of. Okay? Hopefully, if you're married, your skill set complements your spouse's skill set, and you can be an effective partnership to run a home or a family or whatever it might be. Okay? Like I'm a scientist and I'm married to an artist, and she can do all the creative stuff and I can do all of the technical stuff, sort of. She gets to pick what we watch on Netflix. I have to deal with the Wi-Fi issues when the Netflix goes down. You know, we work together great. That's complement, working together. Complement with an I is, oh my, you look pretty thin, which you also have to say to your wife quite a bit, but they mean different things. So when I'm talking about complement here, I'm talking about a protein system that works together as a cascade, as a number of important players. They're referred to as just complement protein 1 through complement protein 9, and we just abbreviate that C1 through C9. So in this picture here, they show what's happening in complement, and all of the proteins are just labeled C1, 2, 4, et cetera, et cetera. Now, I can't quite, well, maybe I can do this. One more. Okay, so I want to start by describing complement by just showing you this part of the picture. Complement protein 3, C3, gets broken down into two pieces called C3A and C3B. C3A interacts with other cells, and I'll talk about that later on. What we want to follow here is C3B. What C3B does is it converts C5 into two pieces called C5A and C5B. And what C5A does, C5B doesn't do anything as far as this figure is concerned. C5A then recruits C6, C7, C8, and C9. And they, together with, I think, some other related proteins, form what's called the membrane attack complex, or MAC. What this is it's a big channel that's built into the cell membrane of pathogens, and this is supposed to represent some sort of cellular pathogen like a bacterium. So this big thing that looks like a, I don't know, birthday cake or something, is inserted into the membrane. These other things over here, which are not quite as colorful, are representing other instances of membrane attack complexes. They're just big channels that are making holes in these cells. And when that happens, whatever's inside the cell spills out. Okay? What's going to happen if we make holes in you and your insides spill out? Yeah, it's going to destroy you. That's exactly what's happening here. By making holes in these cells, it just destroys the cells. It's what's called lysis. So it actually, in this paragraph here, it says causing pathogen to lyse. Lysis is the process. Lyse is the verb form of that word. So sometimes complement is referred to as causing cytolysis or lysis to destroy the cells. Now, I started here because this is the basic aspect of what complement does. And there's actually two ways of describing what complement, or not two ways, there's two aspects of how complement works. Okay, so let me go back over here. So complement can either work through what's called the classical pathway or through the alternate, either alternate or alternative pathway. And I don't really care which it is, but the other pathway. Okay. What I described so far is essentially the alternate pathway, where C3 starts things off. Something has to break C3 down. And what that something isn't important just yet. For the alternate pathway, there are a number of things that act to break down C3 and get it all started. And those are things that are going to be activated non-specifically, just when there is a bacterial presence or something like that, complement to be activated, and it ends up causing the destruction of those pathogens. Now, in the figure here, I just want to point out what they're saying about C3A. It says C3A attracts phagocytes to the infection site. Okay. I really wish they went a little bit further to say what this is because of how this works. Okay. So I'm going to come back to this because how this works is part of the inflammatory response. So when I talk about the inflammatory response next, I'll come back to what this is pointing out. But this is essentially the alternate pathway. Classical pathway is what the whole picture here is showing us. And it's going to have all of the C complement proteins involved. C1, C2, C4, and then C3, C5, and C6, 7, 8, 9. So maybe when I just described that, you said, okay, it starts with 3 and it jumps to 5, where's 4? The numbering here is just historical. It's a matter of when these particular proteins were discovered, they were numbered in order. So the first one that was discovered was C1, and then C2, and then discovered C3, and they said, oh, look, there's another one, we'll call that C4, and then they found C5, 6, 7, 8. Okay. It's a little annoying that they're not exactly numbered in order, but it's more of a historical reason, and that's how naming and numbering things in science often works. So with the classical pathway, these other proteins are involved. C1, which some sources will say C1 is this whole thing here, sort of in the bracket. The label here seems to be pointing just at that green thing, but I think it's really the whole thing. C1 interacts with antibodies. Now, antibodies are part of the adaptive immune response. So that means that this is how complement fits in with the specific response that we're going to get to talking about later. So there's something about the invading pathogen that causes antibodies to bind to it, which is what this picture is suggesting. Okay. And then at the other end of the antibody, there are places where it binds to complement. And when C1 binds to antibodies which are bound to a pathogen, that then causes C2 and C4 to be broken down, and part of each one comes together to form what's called C3 convertase. Now, for the alternate pathway, C3 convertase does not have to be the thing that breaks down C3. Other things can do that when it's a non-specific response. But when it's a specific response, all of these other things are part of the response, part of what's going on. Okay, and so we get to this point here. So when I zoomed in on part of this, and I just wanted to talk about that to begin with, I cut out the C3 convertase, which is half of C2 and half of C4, and C1 and all that. I cut that out because it's not part of the non-specific response that we call the alternate pathway. I did it that way on purpose. I would prefer with complement to just talk about the alternate pathway in context of the innate immune response. And then when we get to the adaptive immune response, then include the C1, C2, and C4 stuff. However, I don't like doing part of it and then coming back an hour or so later and doing the other part. So I want to talk about everything here so that you know that it exists. And we haven't talked about adaptive immunity, specificity, or antibodies yet. But when I do, just remember that this is one of the things that happens. It's a specific response that ends up lysing, destroying the pathogen there. Okay, and that's complement. Okay. Then the next thing to look at, to talk about, is the inflammatory response. Now, you're all familiar with the inflammatory response in the fact that it's all happened, that all of you have had it happen to you. but you probably weren't aware of it or called it the inflammatory response. I'm not crazy about these pictures demonstrating it, but I want to use them in the book to talk about what it is. This is, of course, specifically using wound to the skin as an example. So this is a cut that's gone all the way through the epidermis. So this wavy line here represents the basal membrane of the epidermis, and the cut's gotten all the way down into the dermis. And so bacteria on the outside surface can get in. We have an infection setting in here. And so the inflammatory response is how we deal with that infection, or I should say how we coordinate the immune response, because it doesn't actually have to just be to an infection. They, for some reason, didn't label it in the picture, but next to it where it says the number 1, it says mast cells detect injury to nearby cells and release histamine. So the 2 cells that are sort of on either side of the word histamine, those are mast cells. Mast cells are a type of cell we find in connective tissue. They're not necessarily an immune system cell, although their function to release histamine from inflammation is an immune function. But they're not a type of white blood cell. They're just a connective tissue cell. And they release this signal called histamine. Histamine causes blood vessels to dilate, therefore increase perfusion or more blood flow, and to become a little bit leakier, so that things that are in the blood can leak out into the surrounding tissue. Histamine is going to be released where there's infection or injury, and so it's going to increase blood flow to that location, and it's going to encourage things that are in the blood to be able to move out of the blood and into the tissue to help fight off potential infection or help repair tissue. So in this next picture here, again, I'm not crazy about how these pictures are demonstrating this, but this big red thing here represents things coming out of the blood. Of course, the redness of blood is because of red blood cells, and we're seeing red blood cells here. You would not actually have red blood cells spilling out into the surrounding tissue unless there's a real serious damage to the tissue, which would be called bleeding. But really what it's about is white blood cells moving out. So the graphic artist here, I think, got a little bit lazy in what they did. So these 2 pictures are essentially the same. They just added in the big red spill looking thing here and took out the mast cells and the histamine. But the cells in the bloodstream here, these white blood cells, got copied from one picture to the other, but then also they have the exact same cell. You can tell because the nucleus is drawn exactly the same. So they just took this cell and copied it out here, and took this cell and copied it out here. What would really be going on here is the white blood cells in the bloodstream in the first picture move out, cross the wall of the blood vessel, and get into the surrounding tissue. Now, those are just labeled as phagocytes. They're most likely neutrophils, just because neutrophils are the most common type of blood cell, white blood cell. But they could potentially be monocytes, and if monocytes move out of the bloodstream and into the tissue, they're activated, and we call them macrophages. But odds are they're neutrophils just by sheer number. And once they've gotten out into the tissue, they can then, as phagocytes, gobble up the pathogens and get rid of them, which is what we see happening here. The inflammatory response largely is just this increased perfusion of blood to a site of injury or infection. Now, I want to demonstrate or talk about the inflammatory response in a little bit more classical way. I'm familiar with this because when I was a graduate student, I was in a graduate program that happened to be at a medical school because I was doing biomedical research. And one of my roommates was a medical student, and while he was taking physiology as a first-year medical student, this is one of the things he learned. And this picture was in his notes. His professor used this. And it's a classic, no pun intended, representation of the inflammatory response that's been used probably by medical school teachers for decades. And at the top there, I pulled up a Google image search for this picture and just looked for the biggest one I could fit on the screen. And so the one I grabbed here has at the top the 5 cardinal signs of inflammation. So in terms of training future doctors when they're talking about inflammation, the idea is, what do you see when there is an inflammatory response? But how this picture is laid out actually goes a little bit further to get at that. The central aspect of what's going on here is swelling. When you have increased perfusion, when there's more blood going to the tissue, there's necessarily going to be more water leaving the blood and getting into the tissue, causing the tissue to swell. So that's at the core of all of what's going on. If we go to one side, my side of the picture here, we can see a couple of things that come from the increased perfusion also. We have redness. Now, for the sake of the picture, they show this guy's toga's fallen down and he's embarrassed. Redness associated with embarrassment is the same as redness associated with inflammation, just increased blood perfusion to tissue makes the tissue more red. When you're embarrassed, you're doing it because it's part of an emotional response. But when you're fighting off an infection, you have increased perfusion to tissue. That tissue is necessarily going to take on more of a reddish tint because of that. Now, in the picture I was showing you a second ago from the book, they might have tried to put that red oil spill looking thing in the tissue to represent that idea, but that's not a very good representation of what's going on. Just more blood coming to the tissue is necessarily going to make the tissue a redder color because of the red color of blood as it's passing through the tissue. And then the next thing there is heat. So for the sake of the cartoon, the embarrassed guy who's lost his toga is, I guess, going to pick up the toga with the hand that's also holding his torch, and so he burns his neighbor with the torch as he's doing that. And so that's representing heat. That's just the cartoon aspect of it being kind of silly. But really what it is the heat that makes up our body temperature is largely the result of the temperature of our blood. And so if you have more blood going to a tissue, there's going to be more heat in that tissue. And that's what that's kind of suggesting. But also, we can increase the heat, the temperature of a tissue chemically. There are chemicals that induce temperature changes called pyrogens, pyro as in pyromaniac, and gen meaning to generate. So chemicals that generate heat are pyrogens. What they largely do is they increase mitochondrial activity, and mitochondria, when they're making ATP, are going to necessarily generate heat as a byproduct of that chemistry. Or even sometimes there's evidence that mitochondria can put all of the energy of glucose into generating heat and be completely inefficient as far as energy production is concerned. They won't make any ATP, and they'll just burn glucose down to generate heat. Whichever it is, it's possible for tissue to become hotter, whether it's more blood going to the tissue or the tissue is actively generating heat through mitochondrial activity. Is either one's possible. By increasing the heat, that can be antimicrobial. So earlier I was saying that bacteria that live on our skin have to deal with an acidic environment, and sometimes that's enough to keep cells from looking at getting into our neutral environment from surviving. They're also looking to live at 37 degrees Celsius, body temperature. So if you increase that temperature, that's going to also damage pathogens and hopefully keep them from surviving. That can happen locally, which is part of the inflammatory response, or it can happen systemically, which is what we call a fever. To some degree, you could think of a fever as being a system-wide inflammatory response. Because when you have a fever, you have other things going on which are satisfied by these 5 cardinal signs, but it's not localized to a particular tissue. If we go the other direction from swelling, we see other things, which can also be attributed to the increased perfusion, but instead of the redness and heat, it's more about what's happening to the tissue because of the increased fluid retention. Possibly for the sake of the cartoon, they put loss of function on the outside and pain on the inside. Redness and heat, I could kind of relate to each other in a progression out from the center, but really pain and loss of function, to a large part, are independent or interrelated enough that we can't say one comes before the other. So when there's swelling, there's going to be chemicals released, like I just told you, histamine from mast cells causes that increased perfusion, but also during the inflammatory response, the tissue is going to release other chemical signals, which will bind to pain receptors, and you'll have a sense of pain. Now, pain can be thought of more as a behavioral modification signal. Pain at the site of infection or injury, at the time of infection or injury, isn't probably that important. It's more about driving reflexive responses. So if you step on a tack, for instance, the pain that you feel from that, which is more directly the sharp point on the tack activating the pain receptor. But go with the example. Stepping on a tack, that pain is going to immediately cause you to withdraw your foot so that you don't do further damage. If you cut yourself in the damaged tissue, there's going to be activation of pain fibers, and that can cause a behavioral response. It can be broader than that. Okay? If you sprain your ankle, and a sprain is an inflammatory response, there's not an infection, but there's an inflammatory response caused by tissue damage. If you sprain your ankle is going to hurt, and you're not going to want to put weight on it, which will give your ankle time to heal better. Okay? And it'll also force you to put more of your weight on the good ankle. So again, it's behavioral responses to that pain that's most important. If we think about the systemic fever, it's not just about an increase in systemic temperature. There's also aches and pains and that sort of thing. And when you have that kind of ache and pain feeling, you don't want to do anything. You just want to lay in your bed. And that gives your body a chance to heal up and help fight off whatever infection that fever's directed against. And then loss of function, again, is kind of its own thing, although pain and loss of function can kind of go together. If we think about the sprained ankle, because of the swelling and the pain, you can't bend your ankle as much as you should. Okay, that's loss of function. Or if you have pancreatitis or something like that, itis means inflammation. So an inflammatory response in your pancreas, pancreatitis, is going to result in the loss of pancreatic function, okay, because the pancreas is trying to deal with that. Now, sometimes a problem with inflammation is that the inflammatory response causes symptoms that we want to get rid of, okay, and sometimes we can't. I had surgery on my leg here. When was that? 12 or 13 years ago now. I broke it when I was 15, tib-fib fracture, if you were paying attention last semester. And it didn't heal correctly, and so when I was 20-something, maybe, they put in steel rods to make the bones heal back the way they were supposed to be. And so they put a pin in at the bottom of my tibia and at the top of my tibia. And I have an inflammatory response in my ankle because of that. It's been there for 13 years. I can't bend my ankle nearly as much, my right ankle nearly as much as my left ankle. Okay, so I can point my left toe out quite a bit, and I can't point my right toe out very much at all. That's because of the inflammatory response that came from the surgical intervention. I have sort of swelling of that tissue that's never going to go away. I've lost the function of that tissue because of what's supposed to happen. Now, what really should have happened is I should have had some really good PT after the surgery. But I was in a postdoc then, and I had really bad insurance coverage, so I didn't get the PT I should have gotten. But it's an inflammatory response, and I've lost function. I can't bend that ankle as much as I should because it's sort of chronically inflamed. Okay? And there are a lot of diseases where chronic inflammation is a major player. And it's because this response is doing what it's supposed to, but it didn't stop doing it when whatever it was supposed to help fix maybe got fixed. Okay? Or tissue damage just keeps going on and on. So osteoarthritis, for example, is swelling of the joints because damage of the joints just continues and it never quite gets fixed. And so people have pain and swelling in their joints and loss of function in their joints because it can never quite be fixed. They just keep getting damaged. There are a number of autoimmune type diseases where the inflammatory response is elicited when we don't need it. Okay? When your body's responding to something that's supposed to be there as if it's not supposed to be there. And talking about arthritis, rheumatoid arthritis is that sort of thing. So in rheumatoid arthritis, there's not really any damage at the joints, but the immune system is reacting as if there is, and so you get arthritis from that. And it's just a failure of the system to accurately respond and have inflammation do its job properly. So, there are a lot of things that people suffer from that it's really the immune system doing what it's supposed to be, and the symptoms are largely coming from things like the inflammatory response. So let me back up to the complement thing real quick, because now that I've talked about the inflammatory response, I want to go back to what's going on here with the C3A stuff. Now, what C3A actually does, which is not represented in this picture very well, is that it activates mast cells to release histamine. Okay. And when mast cells release histamine, it causes inflammation. So when there's damage or infection in tissue, the same signals that are going to cause complement to be activated are going to be related to the inflammatory response. And they can use, when the alternate pathway is activated, the first thing that happens is C3 is broken down. And C3A is a chemical that's going to activate the inflammatory response, release histamine from mast cells. When mast cells cause inflammation, there's going to be more perfusion and leakier walls of the blood vessels, and so phagocytes are going to get into the system, into the tissue. And so where it says here that C3A attracts phagocytes to the infection site, it leaves a little bit out. C3A causes mast cells to release histamine, which is going to cause phagocytes to come to the infection site. C3b will be attached to the outside of the phagocyte after it's broken C5 down. It then attaches to the pathogen, and the phagocytes, recognizing C3b, will know which things to gobble up. So C3 really interacts with mast cells to cause inflammation and phagocytes to increase the phagocyte's ability to get rid of either the dead or dying pathogens that are present. So this arrow here is just showing that C3A, or at least C3 in general, interacts with other things. It just leaves it out that it's mast cells releasing histamine, which causes the phagocytes to show up, which is what this picture here is showing. These phagocytes are showing up because of the histamine signal that was released earlier. Just C3A is one of the things that causes mast cells to release histamine. It's not the only thing, but it's one of the things it does. So it ties complement into the inflammatory response. That's the end of that section. So that section, which is again entitled, or I said this earlier, barrier defenses in the innate immune response. It's all in one section, which is basically covering this initial stuff that I mentioned here. Okay. So now we want to move on into the adaptive immunity, and 9:30. Okay. Just making sure I'm keeping an eye on the time. So hopefully an hour and a half is enough time to get through all of this. No, it's not. So I'm going to go real quick. So the way that the book presents this, the way that any book presents this, is always kind of quirky, and I don't entirely like it. So I want to start off by talking a little bit about what the specificity is about. And you've heard about antibodies before, and that's a good example of what it's about. Antibodies are things that bind to antigens, or antigens are things that cause antibody reactions. And in fact, that's what the word antigen means, to generate antibodies. They don't actually generate antibodies. Antibodies are made by a type of lymphocyte. But when an antigen is present, then in your system, you'll see an increase in the number of antibodies. Now, in the chapter here, so we just did the innate immune stuff here. Then there's a section here about T lymphocytes and a section here about B lymphocytes. And for the most part, that's a good progression to deal with the T cells before we deal with the B cells, except for I need to anchor all this stuff to things that you've probably heard of before. And you've heard of antibodies, and antibodies are actually made by B cells. So let me move to this section for a second. And I just want to show you this picture here. That shows us antibodies. Now, this is a ribbon model of an antibody. So this is taking actual data from protein structure studies, and this is what antibodies look like. They're made up of 4 different parts. The green, red, blue, and yellow things are 4 separate pieces of protein, subunits that come together. The green and the yellow ones, which are also represented in the schematic side over here, are short. They're what we call the light chains. And then the blue and the red are long, what we call heavy chains. Now, when we're talking about proteins, heavy and light, long and short are interchangeable. So a heavy protein is going to be a long protein. A light protein is going to be a short protein. So that's where the terminology comes from. Now, you have in your genome genes for the heavy chain and the light chain in antibodies. Okay. And actually, I'm not an expert on this. You might actually only have one gene, and it can be either translated to make the heavy chain or just light chain. But I usually think of it as 2 different genes, and we'll stick with that. Now, within the gene, it codes for proteins that have variable regions and constant regions. The light chain has 1 constant region and 1 variable region. The heavy chain has 3 constant regions and 1 variable region. So let me talk about what variable regions are, and then constant is sort of not that. What we mean by variable region is that that stretch of the DNA is variable from lymphocyte to lymphocyte. When a new lymphocyte is made in the red bone marrow, that sequence in its antibody genes gets randomized. So enzymes come in and chop up the DNA and put it back together. And for every lymphocyte, because it's a random process, they're going to end up with different variable region sections of the gene. Now, a gene is instructions to make a protein, and proteins have three-dimensional shape. So when the gene is variable, then the corresponding protein section can be variable from one lymphocyte to the next, which means it'll have different three-dimensional shapes. And really what we're talking about is this is the variable region of the light chain, and this is the variable region of the heavy chain. Where they come together from one lymphocyte to the next, they're going to be different. So every lymphocyte makes its own type of antibody. At the end of the variable regions, we have what are called the antigen binding sites. That's what's binding to the pathogen or whatever it is that's causing the problem. Sort of like a lock and key, however, most proteins interact together, or a neurotransmitter binding to its receptor, or an enzyme binding to its substrate, any of those kinds of things. The antigen binding site binds to the antigen. And it's a three-dimensional puzzle piece kind of interaction. The antigen binding site's only going to bind to a particular antigen that fits its shape. Now, the variable regions are randomized, and every lymphocyte makes its own type of antibody, which means that at random it's making antibodies that can bind to all sorts of different three-dimensional shapes. We don't know what we're going to be introduced to in our lifetime, the things that can potentially make us sick. So the antibodies are just making every conceivable shape to be able to interact with any conceivable antigen. Now, there is an upper limit. There's a finite number of things that we can make in our body. So we aren't necessarily going to be prepared to make every possible antibody deal with every possible antigen that will ever meet in our whole lifetime. But the vast number that we're dealing with, we're going to make a bunch of things that we can. Now, there are some things, and I don't entirely know how the process works, but there are some things that we're going to naturally develop antibodies to, or naturally make some. So just in the random process, there are some things that everybody's going to be making antibodies to. Things that we as a species probably have to deal with on a regular basis. So we've adapted to it in an evolutionary sense. So that's the variable region at the end of either of these. An antibody has two antigen binding sites. It can actually bind to two things. So if you have two bacteria, and each one's bound by one of these antigen binding sites, and they're going to be linked together. Okay? And so one thing that antibodies can do really well is they can clump up a bunch of antigens together, which can make them ineffectual. Now, I wanted to talk about antibodies because you've heard about them before. But they're not the only thing that's part of the adaptive response. So let me go back to the T cell portion because that's where the other thing comes from. And T cells have what are called T cell receptors. And here's the antibody we just looked at. It's got 4 chains giving us 2 antigen binding sites with variable and constant regions in them. This is a T cell receptor. It's only 2 chains called the alpha chain and the beta chain instead of heavy and light. They're roughly the same size proteins because they're both anchored into the cell membrane. But they have a constant region and they have a variable region. And really, if you want to think about it, the transmembrane region is constant also because these receptors always have to attach to the cells in the same way. So there's this little variable region on the outside, and then the rest of it's pretty much fixed for how these cells, how these proteins work. They aren't pointing it out here, but at the end of each of these, where the variable region is an antigen binding site. So just like in the antibody here, at the end of the variable region, that's where it has the 3-dimensional protein structure that makes it interact with its antigen. There's only one of them here. So T cell receptors don't cross-link multiple things together. They only ever interact with one thing at a time. But it's the same kind of idea. In the gene for the alpha chain and in the gene for the beta chain, the variable region DNA gets randomly sorted so that each T cell makes its own type of T cell receptor, just like each B cell makes its own antibody. I didn't say that before, but the antibodies are made by the B cells. Hopefully it was obvious because I was showing you that in the B cell section of the book. So for the adaptive immune system, we're talking about how B cells and T cells, or B lymphocytes and T lymphocytes, interact with specific pathogens. B cells do that through antibodies, T cells do that through T cell receptors. At the molecular level, they work on the same principle, that they have this variable region that interacts with specific antibodies. Those are made randomly so that the cells potentially can be able to recognize anything out there. Okay. But those are the players that we're talking about. So here at the beginning of what I have here, antigens and antibodies and T cell receptors, the reason I put these in here at this point is this is the basis for specificity. This is how these cells interact with what's in our environment. Okay. So I could just as easily, for the second item there that says T cell receptors, I could have said antigens and T cell receptors, because T cell receptors are what T cells use to interact with antigens. Antibodies are what B cells use to react with antigens. Okay. So the next thing I want to talk about is the life cycle of a lymphocyte. Okay, so this is talking about both B cells and T cells, but it's important to think about them first off collectively. Now, again, the book breaks this down into the T cell section, which is where we are right now, and the B cell section. So I'm going to talk about this mostly in regards to T cells, and then when we get to the B cell stuff, I'll point out what fits in there too. But what's important here is mostly about the T cells. T cells are the big players in what I'm getting at here. Now, I said this section is the life cycle of the lymphocyte. Lymphocytes are white blood cells. Blood cells are made by bone marrow, red bone marrow. So like every blood cell, lymphocytes are made in the red bone marrow. The difference between B cells and T cells is what they do next. B cells stay in the bone marrow, and we're going to use the B in bone marrow as a mnemonic to remember that B cells are bone marrow cells. The B in B cell actually stands for something else that doesn't have anything to do with humans, but for our sake, we'll call it B bone marrow. And then T cells move to oh, shoot. I thought there was going to be a picture of it here. T cells move to the thymus. And in fact, the T in T cell stands for thymus. So last week when I was talking about the lymphatic organs, remember I made the point of there are primary lymphatic organs, or lymphoid organs, and secondary lymphoid organs. The primary lymphoid organs, or tissues, are bone marrow and the thymus. And they're primary because that's where lymphocytes go through this process. Okay. Everything's born in the bone marrow. B cells stay in the bone marrow, T cells move to the thymus. And then the next stage of the lymphocyte life cycle is their maturity. They go to school. And the T cells, when they go to school, they have two major tests that they have to pass. And actually, let me get away from the book for a second. They have two major tests that they have to pass. One is called negative selection, and the other is called positive selection. Now, in negative selection, the lymphocytes tested to react with immune system components. And namely, it's what's called MHC. And I'll point that out in a second when I get back to the book. And then positive selection is when the cells tested for reaction to self-antigens. Oh, and I should add in why they're called negative and positive selection. Negative results cause cell death, and here, positive results cause cell death. So, both of these things have to do with T cells. B cells only have to go through one of them. But negative selection is when T cells are tested to be sure that they react with this other immune system component called MHC, which I'll show you in a second. If they cannot do that, if they have a negative result, they are destroyed. The T cell can't react with MHC. It's not a T cell. It's not a useful T cell. So we get rid of them. Positive selection is when the cells are tested to see if they recognize things that are supposed to be in your body. If it does recognize something in your body, you destroy it. Okay? So positive results cause cell death. Now, we've actually already thought about an example of this, and that has to do with blood type. If you are blood type A, that means that your red blood cells express the A antigen on their cell surface. So during positive selection, the T cells, or B cells for that matter, are tested to see if they react with the A antigen. If they do, then they're destroyed. You don't want lymphocytes that react with your blood cells. If they don't react, then they survive. So people with type A blood destroy lymphocytes that make antibodies or T cell receptors that react with the A antigen. They do not destroy things that make antibodies or T cell receptors that react with the B antigen. So type A blood people will have antibodies against type B blood. And conversely, type B people will destroy lymphocytes that recognize the B antigen and keep the ones that don't, which would include ones that make A antibodies. Okay. So the antibodies present that are opposite blood type really are just about this aspect of things. You're tested to see if you recognize self-antigens. If you do, you're destroyed. Because we don't want autoimmune reactions. It's not a perfect system. Autoimmune reactions can occur, but they are fairly rare because most of this gets rid of it. Now, this happens in the first 6 months or so of life. So a newborn does not have an active adaptive immune response. And last time I was talking about the naturally acquired passive immunity. That's the newborns getting antibodies from the mother through the placenta, and until this is in place, new antibodies that are made can also be passed through breast milk. But once the lymphocytes, whether they're T cells or B cells, are tested for this, and they won't react with self-antigens, then the adaptive immune system is ready to go. A lot of this happens in the first 6 months of life or so, but some aspects of it might take a little bit longer, a couple of years or something like that. So usually the immune system isn't considered fully functional until a toddler's 2 or maybe even a little older than that. So now I want to talk about what MHC is. And this is one of the places where the book uses dendritic cells, unlike a lot of other sources. What it's saying here is fine, and And there's a couple of reasons why it's doing this. So like I said earlier, it's partly so the book sort of sets itself apart from other sources. Nothing that it's saying here is incorrect. I just think it's making a little bit more of dendritic cells than are probably appropriate. The other thing that it can do here is in one picture show you two things, which every other book has to show you in two pictures. And oftentimes those other books don't get the second picture in. As some editor somewhere says, we don't need the second picture, we got the first picture. So here, this picture shows you two things that are important to talk about. Now, antibodies or T cell receptors deal with antigens. Antibodies are about dealing with unprocessed antigens. The antigens are just floating around in the bodily fluids in your body. T cell receptors are on the surface of T cells, and they deal with what we call processed antigens. Antigens are processed because of how they interact with cells in our body. Now, phagocytes, whether they're neutrophils or monocytes, whatever, when they ingest something, which is represented in this picture here, and I showed you the video earlier of the neutrophil chasing down that bacteria. Once it's ingested something, which is represented in this picture here, that thing that's been ingested is contained within a vacuole called a phagosome, which I don't think is labeled here. And that phagosome merges with what's called a lysosome, which is an organelle that every cell has. It's basically the stomach of the cell. It has digestive enzymes and acids in it. It has a low pH. So when that low pH digestive enzyme organelle merges with the phagosome, it digests the phagocyte. It digests the pathogen, which is what's shown here. So all these little light blue spots represent things that are breaking down the pathogen. And then the pathogen, when it's broken down here, will be the bits and pieces that are left over of the pathogen will be paired up with what's called MHC proteins. Now, MHC stands for something. What it actually means is relevant to immune function, but it's not the whole picture of what MHC does. So we're just going to call it MHC. And since MHC was first discovered and named, we've discovered that there's a lot of things, some of which has nothing to do with the immune system. So it's generally just referred to by those initials, and we don't think too much about what they actually stand for anymore. Now, in the picture here where they're labeling this green thing here, which is MHC, they're telling you that it's MHC2 protein. There's two types of MHC, and they're just called type 1 and type 2 MHC. Type 2 is used to bind to pieces of antigens that have been digested by phagocytic cells. And when MHC2 and the digested pieces of the pathogen are together, they then get presented on the surface of the cell. And so phagocytes, neutrophils or macrophages, are referred to as antigen-presenting cells because they gobble up the thing that can potentially make us sick or diseased or damaged tissue, whatever it is that they're getting rid of, break it down, and then present pieces of it on its surface, kind of like really gruesome hunting trophies or something like that. As an antigen-presenting cell, it's then going to be able to interact with T cells, which we'll get to in a little bit. Now, the other stream of what's going on here is the thing that I have the most problem with using dendritic cells to show. Now, dendritic cells are these kind of specialized antigen-presenting cells. So they present more than just the kind of antigens that other phagocytic cells will present. What's represented here is better thought of as happening in other cells of our body. So for a little while, let's pretend that we're not looking at a dendritic cell and ignore the part that I just showed you, because this is a separate process. So let's imagine that we're looking at any cell in your body, okay? and for the sake of what I've already talked about before, let's say it's an epithelial cell from your respiratory epithelium. When it's been infected with influenza virus, it's going to start making copies of the influenza virus. What happens in a cell, and is kind of represented here, is bits and pieces of whatever it's making get paired up with MHC proteins, but instead, and unfortunately the cartoon doesn't draw them differently, but this is MHC1. So any cell, when it's doing what it's supposed to be doing, will occasionally put little chunks and pieces of the proteins that it makes on its surface using MHC1. So fibroblasts will do that with chunks of collagen or elastin or the other fibrous proteins they make. Red blood cells can do that with the blood typing antigens, although that's kind of redundant. Neurons will do that with voltage-gated sodium channels or acetylcholine receptors or whatever it might be that they make. Those are things they're supposed to be making, and they'll put them on their surface. The thing is once somebody's immune system is up and running, those self-antigens don't matter because you've already gotten rid of all of the lymphocytes that recognize those self-antigens. It's the cells in your body that present antigens on their surface that are not supposed to be there. So a virally infected cell, when it's making new viruses, will put little pieces of the viruses that it's making on its outside surface, which is basically a signal to say, hey, I've been infected with the virus. Or if a cell is going along doing what it's supposed to be doing, making some protein, and then something happens and the cell changes how it functions and it starts making a different protein or making a protein wrong, it'll put pieces of the wrong protein on its surface, which will then be a flag that that cell's done something wrong. Now, I want to use an example for something that we've talked about already in the course. Whether what I'm about to describe is actually how it happens or not, I don't really care. I might be wrong, but the concept is solid. Okay. So we were talking about in the pure autonomic failure case study for the autonomic system. The cells that are dying off because they have this alpha-synuclein toxicity. The cells normally make a different form of synuclein. Let's call it beta-synuclein, which might actually be true. But something happens and the cell starts making the protein wrong. And so it's no longer making beta-synuclein, it's making alpha-synuclein. So all along the cell, while it's been making beta-synuclein correctly, has put beta-synuclein on its cell surface to say, hey, look, I'm doing what I'm supposed to be doing. But as soon as it starts making alpha-synuclein, the wrong protein, and starts putting that on its cell surface, that's not an acceptable self-antigen, so that's a clue that that cell's not functioning anymore. So that's an example of something that we've talked about before. Whether that's actually what's happening or not isn't important, but go with the story. Another way of thinking about it, cancer cells, when they're no longer functioning the way it's supposed to be, like melanocytes in the melanomas, if they start becoming cancerous, they're going to be making a lot more melanin, and therefore they can be presenting a lot more melanin on their surface, which will be a signal that something's wrong. Again, I don't know if that's exactly how it works, but I'm trying to use an example that you're somewhat familiar with. So this second pathway here, although it's not represented in this picture as an internal protein being packaged up that way, that's in fact what's happening with any other cell except for a dendritic cell. And so down here at the end, these two arrows converge. And so what we see on the surface of this cell, the way it's drawn, we don't know if this is MHC1 with this antigen or MHC2 with this antigen. But whatever it is, it's presenting the antigens on the outside. So for T cell
development. Okay. This little flow chart shows you what's happening. And this is actually where the book is dealing with the negative selection and positive selection stuff. This is just about T cells. But some of this applies to B cells also. So the cell is born in the bone marrow. Okay? And the thymocyte or T cell, T cell, is fairly undifferentiated. We couldn't look at it and say, oh, that's a T cell versus any other type of lymphocyte. But we know where this cell is going. So it's born in the bone marrow. Being a thymocyte or a T cell, it moves to the thymus. And when it gets to the thymus, it has to go through the positive selection. Oh, I said that wrong, didn't I? I'm sorry, I got these backwards. Okay, I just got those two terms wrong. In positive selection, failing the test causes death. In negative selection, passing the test causes death. Sorry about getting that wrong. Whoops, there we go. So positive selection. The T cell is presented with cells that have MHC on their surface. Okay. And if they correctly interact with MHC, meaning that they're going to be able to do their job as a T cell, they pass. If they can't bind to MHC, then they fail the test and they're eliminated. Okay. That's what positive selection is. Sorry, I got the term wrong. Then the T cell goes on to negative selection, which is still happening in the thymus. Now, I haven't really pointed out because, again, I'm not an expert in this stuff, but in parentheses next to thymocyte here, the early one, the undifferentiated one, there's nothing going on. But then they call it CD4 negative, CD8 negative, which means that it doesn't have that stuff, which isn't effectively any different from this. But then, after passing the positive selection test, the thymocytes, the T cells, will become CD4 positive and or CD8 positive. Now I say and or because, again, I'm not an expert, and I was always in the impression that the cells became either one or the other. But in the book here, it's suggesting, for the sake of this developmental process, they become positive for both things. Now, what CD8 and CD4 are, those are specialized receptors that play a role in how the T cell interacts with the MHC antigen complex. So once they have proven themselves that they can deal with MHC, they then start to express the CD8 or CD4 receptors so that they can do their real job even better. Then they go through negative selection. And with negative selection, the T cell expressing the T cell receptor and the CD8 or 4 receptor is presented with self-antigens. And in the picture, they're showing this with dendritic cells because dendritic cells are a player in that process. But it could be from anything. It could be a red blood cell or something like that, just as easily. If the cell binds to the self-antigen, that's a bad thing because that would mean that it would cause an immune response to that antigen that's supposed to be in your body. So anything that passes the test by reacting with the antigen is destroyed. And if it can't bind to the antigen, if any T cell that makes a T cell receptor that doesn't recognize a self-antigen survives. And then at this point, the book is saying they differentiate into either CD4 positive or CD8 positive cells. And all that means is at this point, after they've gone through these developmental processes, the cells express either one or the other. And what's important is CD4 will only interact with cells that present antigens on MHC2. And CD8 will only interact with cells that present antigens on MHC1. The only way I can ever keep that straight in my mind and say it correctly, and I don't always say things correctly, but this one I'm good at because MHC1 goes with CD8. So the lower number 8 MHC goes with the higher number CD protein. And CD4 goes with MHC2, lower number CD, higher number of HC. It's the only way I can keep it straight in my mind. And after they've gone through all of this and they've differentiated into CD4, CD8 positive cells, they move from the thymus into secondary lymphoid organs, which for the most part would be lymph nodes, but it could be a tonsil or anywhere else. CD4 positive cells are called helper T cells, which it says right over here. CD4 receptor, those that express the CD4 receptor become helper T cells, or T subscript H cells, while those that express the CD8 receptor become cytotoxic T cells, or T subscript C cells. Okay. Now, just because they're mature doesn't mean they're ready to do their job yet. They've just gone through the developmental process. They're ready to go. Now they're going to wait until they meet their actual antigen. Remember, everything that's passed through negative selection is something that does not recognize self-antigens. So it has to wait until some other antigen shows up, okay, until you get infected with a pathogen or whatever it might be. Okay. They're ready for that. But though they're mature, they're not entirely ready for their job because there's a little bit of on-the-job training that's required. So I like to think of what's going on in this picture as kind of like going through college, but then after you finished college and you graduated, you got a little bit more to do. So at the end of this picture, they're called mature cells, but actually in the next picture, they're called naive cells. Okay? Now this picture is picking up the story for the cytotoxic T cell. So the cytotoxic T cell, which is expressing the CD8 protein, will interact with cells that are presenting antigens with MHC class 1 or MHC1. And when all of that fits together, T cell receptor with the antigen on MHC1, which is recognized by CD8, then the cell is no longer naive and it moves into what we call activation. When cells are activated, they proliferate and differentiate. Proliferation means to divide and make copies of itself. So this one cell will divide and make a bunch of copies of itself that make the exact same T cell receptor and therefore recognize the exact same antigen. And they also differentiate into the active cytotoxic T cells and memory cytotoxic T cells. In this picture, a kind of unfortunate cartoon representation of it, the activated T cells go out and destroy cells in your body that have been infected with whatever pathogen they're sensitive to. Memory T cells don't do anything. They hang out and are ready for future infections. So if you get infected with the influenza virus, let me use chickenpox as a better example. You get infected with the chickenpox virus, you get the chickenpox, and you fight off that infection, and the chickenpox go away because cells that are sensitive to the chickenpox virus, no, I shouldn't say it that way. Cells that are that make T cell receptors against chickenpox virus. Once they get activated, and how long it takes to get activated is kind of similar to how long people have the pox. Once they're activated, they go out and they find the cells that have been infected with the chickenpox virus, and they destroy those cells. But the memory cells don't play a part in actively getting rid of that infection. Instead, they're around so that in the future, you get exposed to chickenpox again, you're already immune to it, and you can get rid of it. Now, shingles is a different story. if you've had the chickenpox, as long as it wasn't a very mild case, you're not going to get the chickenpox again. You've built up an immunity. You have that immunity because when you were exposed to chickenpox the first time, you made memory cells, memory T cells, memory B cells, sort of thing, and you have that immunity.
Just a quick question on that. My cousin just started a job in a hospital. She had the chickenpox already, but it wasn't properly marked. So to give her the vaccine, they infect her with the virus again, and she ended up getting chickenpox again. She currently has chickenpox, like 22. She's getting a little.
Poor woman.
Is
So I qualified what I was saying by if you didn't have a mild infection. So if she had chickenpox before, but it wasn't a full-blown infection, then she probably didn't develop an immunity to it. Or a very effective one. So her titer against the chickenpox virus was probably too low to be able to be in there. And so she got the Varivax vaccine, which is not living chickenpox viruses. Not that viruses live at all, but not virulent chickenpox viruses. But she probably had the virus in her system somewhere, and so the vaccine probably got that stuff up and going, and she went through another round of having chickenpox. I believe that's probably what happened. Okay?
Would it be worse the second time around?
Well, if you get chickenpox the second time around, that means the first time around was not very bad, because it wasn't bad enough to get the immunity. So yeah, if you get a second time around, it's probably going to be worse the first time around. Also, chickenpox worse when you're older. It's best to get chickenpox as a child. Now, a little quirk about chickenpox, which has to do with shingles, is that when you go, when you build up an immunity to chickenpox, you do not actually get rid of all of the chickenpox virus. Now, what I'm showing is cells that have been infected with chickenpox, but you don't actually clear out all of the chickenpox virus in your system. Chickenpox viruses hide out from your immune system in mainly dorsal root ganglia. So remember, in the peripheral nervous system, there are these ganglia that are just outside the central nervous system called dorsal root ganglia. And they are not in the central nervous system, so they're not behind the blood-brain barrier. But actually, the glia and ganglia do have sort of their own version of a blood ganglion barrier, if you will. So it's a slightly protected area, and the virus can hang out there. And then later on, if something happens, the virus will migrate through that nerve for the dorsal ganglia to extend and move out to the skin and cause pox-like marks on the skin, which is what we call shingles. So what I'm talking about here, getting rid of the infection in chickenpox, you don't actually get rid of the infection. You can have the virus on hand, and in the future you might have a flare-up, but only where that particular nerve is located. So the pox, if you will, or shingles, are going to be located to just the part of the skin that nerve's connected to. It's a way for the virus to get around our way of getting rid of the virus. Liz, did you raise your hand?
It was the shingles question.
Okay. So yeah, the shingles thing is a little quirk in the chickenpox story. But other than shingles, what I'm talking about here is pretty well represented. I could have done it with influenza. The thing is that influenza viruses mutate at a pretty high rate. So you get the flu one year, the next year you can still get the flu because the virus has mutated enough that the memory cells are probably not going to recognize the new version of the virus. So the influenza is not a great example for this, but the chickenpox vaccine doesn't, I mean, the chickenpox virus doesn't mutate like that. So once you're immune to chickenpox, you should be immune all the time. It's just that the crafty little buggers actually don't completely leave your body. I said that they're found in dorsal root ganglia. In fact, they find ganglia somewhere in your body to hang out in. And where you have shingles usually corresponds to where the ganglia are, because it's where the nerves are connected. So if you have shingles anywhere from your neck down, it's because of dorsal root ganglia, and it's going to be limited to the part of the body that that particular nerve serves. If it's from the neck up, then the virus got into a cranial nerve ganglion, and it's going to be limited to where that particular thing is. And I happen to be pointing out my hairline here because my wife's grandmother got shingles right at her hairline, which means that it hung out in the trigeminal ganglia and actually moved through the ophthalmic division of that nerve to right here. That's the problem. My neighbor had shingles once too, and based on how he got those shingles, we know that the virus hung out in the, oh, I'm probably going to get it wrong, but I think it's the nodose ganglion, which is a weird name for a ganglion, but it's a ganglion that's related to nerves that go to the pharyngeal muscles, okay? Which means he had shingles in his throat. The reason I know this story is because I hadn't seen him for a while, and I saw him outside. You know, when we're doing yard work, we usually interact a little bit. And so I saw him and I said, "Oh, Bob, you look great. You've lost a bunch of weight." And he goes, "Well, actually, I had shingles in my throat and haven't eaten for 3 weeks." So not the best weight loss plan in the world, but he was a big guy, so it made him look a lot better. But of course, it was because he was sick. But yeah, so wherever the virus happens to hang out, when it evades the lymphocytes are going to try to get rid of it, it'll come back possibly later in life as shingles. Just because you have chickenpox doesn't mean that you're necessarily going to get shingles. It usually has to be some sort of some sort of stress trigger. My wife's grandmother, it came on because she developed cancer. So there are a lot of things going on. I don't know what triggered Bob's shingles, but something did too. Anyways, so this is, again, called a naive cell here, whereas at the end of this process, it's in the book, it's called mature. It's the same cell. So this cytotoxic T cell here, this CD8 cell here, is this cell here. And the first time it interacts with whatever antigen it's specialized for, it proliferates, makes a bunch of copies of itself, and differentiates into the active and the memory types of cells. Now, there are situations where this doesn't quite work exactly this way. With the book using the dendritic cell as the example of the antigen presenting cell, this is pretty much how it worked, because part of the activation process, going from the box to all of the cells at the bottom, requires a chemical signal, a cytokine, to tell the lymphocyte to proliferate and differentiate. The dendritic cell can do that. The dendritic cell can say, yes, you found the right thing, here's some cytokines, go forth and multiply. But if the antigen presenting cell isn't a dendritic cell, if it's a body cell that's been infected with something, which is another source of this, sometimes that infected cell isn't able to release the signal. So the activation process needs a little help. Where do you think it gets help from? And I'm emphasizing the word help for a reason.
Helper T cell.
The helper T cells, yeah. The reason they're called helper T cells is they help activating the other types of cells. In the book, they highlight the fact that they help B cell activation, but in fact, they can help cytotoxic T cell activation too sometimes when it's necessary. Whoops, there we go. So here's a similar picture that's dealing with the helper T cell. Okay. So subscript H for helper T cell. It has a CD4 protein and it interacts with antigen presenting cells that have MHC2. Okay. The antigen presenting cell will definitely be capable of helping to activate the helper T cell. But an activated helper T cell then releases cytokines. And those cytokines can be part of helping cytotoxic T cells or B cells become fully activated. Now, this is a weird picture. What I just described is just the A part of this picture. The B part of this picture is actually not at all related to what I just talked about. The B part of this picture is really what, which this picture here is showing. Okay, so C cells, when they're activated, they go out and kill things. Cytotoxic means killing cells. That's what they do. So at this point, having talked about cytotoxic T cells a little bit, I want to go back to this video, which is cytotoxic T cells on patrol. Okay, so we already looked at this video because it's a beautiful representation of how white blood cells move, which is true of phagocytic cells and lymphocytes. But the rest of what this is going to get into is how this cell here kills this cell here. So this is the cytotoxic T cell, like what we were just watching. And then this blue cell is a cancerous cell, something that's not functioning properly, so it's presenting on its surface antigens that mark it as a bad cell. And so it's going to be eliminated. So let me play this, picking up from, okay, so this is the end of what we were watching a second ago. And let me finish out this video here.
When a T cell encounters a cancer cell, an explosion of membrane protrusions explore the surface of the target. Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized in the rear of the T cell to the cancer cell. These toxic granules secrete specifically for the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate. Now we'll leave you with a few more movies of T cells doing what they do best, killing cancer.
Okay, so that just plays a few more examples of this. But so let me back up and explain what he's been saying here. So this is where we started. Again, this is the cell in your body that's not doing what it's supposed to be doing. And the cytotoxic T cell is going to kill it. So, oh, shoot. So first they're just showing that using the same sort of mechanisms for movement, once it's found its target, its cell membrane reaches out and finds or explores the cell to get more information about it. And then we switch over here, and actually let me back up. Didn't make it. Okay, I'm not going to be able to catch the frame I want to perfectly. Come on. I'm trying to capture the frame, and I can't quite hit it perfectly. There were red granules in the cell. Now they're using a dye that highlights where the toxins are that the cell is going to use to kill the cell. They're kept in kind of the tail of the cell until they're needed, and then they move up, which is what's happening at this point, to be available to be injected into the cell. Okay. Oh, there we can see it. And so this is just an image, pretty well rendered picture of 3 cytotoxic T cells and 1 cancer cell here. And again, the red are these granules that contain the toxins. And they spin around a little bit so we can get a 3-dimensional image of it, and they pull the blue part out of the picture. So we're just looking at the T cell. But those are these granules of toxins in the cell. That end up getting released in. There we go. So rotating around, we can see they're inside the cell there. One thing that was said at this point was something to the effect of not affecting innocent bystanders or something like that. I don't remember exactly what the voiceover says. The cytotoxic T cell is going to latch on to the cancer cell and only inject the toxins in where there's contacts. In the pictures in the textbook, this is always depicted very differently. These pictures, because a graphic artist has just copied what they drew once and pasted it over and over again, this is not really what these cells look like at this point. In the video, we can see they're very abnormally shaped cells. They're reaching out and they're surrounding the cell. In the book, they always want to highlight where the receptors and CD proteins are so we can see that connection. But really, that would be happening in the connection between the cytotoxic T cell and the target cell. And it doesn't just blow the cell up like this, because if this is what happened, then what was inside the cell can get out and possibly affect the neighboring cells. So these are always misleading pictures. The video here is a better representation of what's going to happen. And then pretty much the rest of this video is just showing where this is a cancerous cell. It's no longer blue, it's now red because they're using a different stain. The stain they're using here, when it gets, when the cell gets injected with these toxins, is going to make the cell flash a bright red briefly when it first happens. And so that's what we see happening over and over again here. So there's a T cell moving in, and it injects the toxins and the cell lights up. Here it is happening again. There we go.
Does that mean that it's dead, or?
I'll talk about that in a little bit more detail in a second. What exactly that means. It's going to die. It's not dead just yet. Now I played this out for a little while because I knew there was one where there's 2 flashes, which okay, actually I showed 2. So a second cell latched on, and so there's the first flash, and then this cell, nope, that cell does again. So it's sort of like multiple doses of what's being injected into it. It's killing it. And so a thing about what's happening in killing it. Let me go back to here where I can write this. The cell death that we're talking about here, which actually right up here I write cell death, and I'm still talking about the same thing. The cell death is what's called apoptosis. Now, this word, apoptosis, looks like it should be pronounced apoptosis to a lot of people. The second P is silent. Okay? Apo is a prefix, and tosis is the root word. Okay? And in tosis, the P is silent, so it's apoptosis. Some people do, in fact, pronounce it apoptosis, which kind of bugs the hell out of me. But this is not a class about how you pronounce words, but that you understand what they mean. What apoptosis is it's more appropriately than just calling it cell death, it's programmed cell death. Okay? cells in your body die on a regular basis. They do it on purpose. Sometimes they do it like a red blood cell that is cleared away by a macrophage. Sometimes they do it because they're no longer needed. Or sometimes they do it because it's an important part of especially development. Your fingers exist because of apoptosis. When you were 7 or 8 weeks in utero, your hands were webbed flippers. All of the tissue that's no longer part of your hand was made up of cells that went through apoptosis. And they died so that you had discrete individual digits. Organisms that have flippers, like fish, or bats, they're not flippers or wings, but any organism that still has tissue in between the bones and the digit didn't go through that process, and they didn't go through it on purpose because it's part of how they develop. In our development of our hands, all of the cells in here went through apoptosis. Now, what apoptosis is it's the clean way for a cell to die. It usually starts with DNA breakdown. There's a better term for that, but I'll just use breakdown. And then organelle decomposition, and then cell fragmentation. Maybe fragmentation is the word I was thinking about for DNA, but I'll stick with breakdown. So DNA is normally a big molecule, but at the beginning of apoptosis, what happens is the DNA breaks down into smaller and smaller pieces. And in fact, that's often the signal that starts apoptosis. If anything starts to break down your DNA, then the cell's going to die. The organelles inside the cell will start to break apart. They might just dissociate or something like that. And then the cell itself will fragment. What does not happen is a cell bursting open. The point behind this clean apoptosis is to leave a nice corpse. If you're going to die on purpose, and I'm not suggesting that you do this, but if you're going to commit suicide, it's best if you do it in a clean way. Okay? This is very gruesome, but it's the only way to really get this point across. So,
Dr. Kevorkian.
So you can take sleeping pills, which actually isn't that clean because you usually vomit the hell up, but you get in a bathtub and you slit your wrist or something like that. It's a lot easier to clean up than if you use a shotgun. Okay. Again, I'm not saying that you should try to, but I'm just trying to illustrate the point. So in apoptosis, these are cells that are dying, but leaving behind something that's very easy for phagocytes to clean up after. Necrosis, which is the opposite of that, is very dirty. That's where cells just burst open. Another way of saying necrosis is lysis, which is what we meant when we're talking about complement. So these are cells that don't lyse. They don't break open and spill their contents. Instead, they fragment down into little chunks that can be cleared away very easy by phagocytes. Now, what we were looking at in the video there are toxins that cause this to happen. And that flash is probably before the DNA started breaking down. Just the indicator dye that's been put in those cells reacts with the toxin itself. So when the toxin is injected, the cell flashes for a brief second. But what's going to happen after the flash is the DNA is going to break down, and eventually the cell is going to fragment apart. And then phagocytic cells can clean up the debris. That's basically what apoptosis is. Now, really in the section above, a little bit of it's no longer on the screen, but the positive selection, negative selection, both of which end in cell death. In the picture in the book, they just had a red X saying no longer a cell. But really what happens is that cell goes through apoptosis and is cleared away by phagocytes. apoptosis is a fairly common process in cells, and it's very important to be sure that we have the cells that we need versus the cells that we don't need in our body. Now, sometimes the cells that we don't need die a horrible death and can create other problems, but when we can control it, which happens a lot during development and can happen when cytotoxic T cells or natural killer cells can play a part too. They can inject toxins that trigger apoptosis. Okay? Sometimes you can't cause apoptosis. Sometimes the cell's so far gone, the only thing that you have at your disposal is a shotgun. And so earlier when I was showing you the natural killer cell, I highlighted that there were two bold-faced words talking about how natural killer cells do their job. The second of those bold-faced words was granzyme. And granzyme is probably what we saw in that video there. Granzyme goes in and breaks down DNA, which causes the cell to granulate or fragment up. And that's where it gets its name, granzyme. The other one in there was perforin. What does perforin sound like it does? It perforates, okay, which is it makes holes in cells. So if you can't induce apoptosis in a cell, which is a clean way to destroy it, then perforin will blow it all to hell, okay, which can cause problems. If it's an infected cell, it's got viruses in it, and you have to perforate its membrane, then the viruses are going to get out and possibly cause further infection. That's not the best solution, but sometimes it's all that's left to get rid of the cell. Because if you don't destroy the cell, then those viruses are just going to continue to multiply, and the infection is going to get worse. But that's sort of what's happening with that at that point in the video where we see the flash. So I mentioned the helper T cells. They help activate things, and they talk a little bit about that here. Shoot, I thought it talked a little bit more about the cytokines. Oh, there's a table, that's why. So cytokines are just a general name for a bunch of different things. And an important group of cytokines are called interleukins, which is abbreviated IL. Interleukin means between white blood cells. Inter for between, and leukin as in leukocyte. So ILs are signals that white blood cells use between each other. And so here they're mentioned when helper T cells help B cells, they use interleukins. And here in the book, they highlight 4, 6, 10, and then they say in others. Other sources I've seen usually say that interleukin 2 is the most common interleukin used for B cells. I don't know if, since I learned that, the information's been updated or what, but other sources I've seen, IL-2 is the more common signal that helper T cells use. So here we have listed 3 different types of cells. We got the helper T cells, cytotoxic T cells, and regulatory T cells. Helper T cells are the CD4 cells. Cytotoxic T cells are the CD8 cells. So we've talked about both of those. Regulatory T cells are something new. Now, they have a CD protein that they're associated with too, which is not important right here. What I want to say is regulatory T cells, which are called Tregs or suppressor T cells sometimes, are what we heard about in the podcast as the lullaby cells. Okay. So these are the cells that slow down the immune response. And in the information I was talking about in regards to hookworm, a parasitic worm infection might actually caused coevolution of this component of our immune system to keep things under control. And the hygiene hypothesis is really the fact that we're removing pathogens that cause regulatory T cells to be more active might mean that we're removing a regulatory aspect of our immune system, leaving the attack cells, which are largely cytotoxic T cells, free to roam our bodies unimpeded. And so if they don't have anything to actually react with, they might say, hey, look, pit dander, let's have some fun with that. Or, hey, look, grass pollen, let's have some fun with that. Whatever it might be. So those are just examples of, simple allergies. But those sorts of things might be just a result of the lack of regulatory T cells keeping the cytotoxic T cells in check. And that's the core of what the podcast was getting at there. And so I wrote those terms up on the screen. So that's the end of this section here on the T cells. The B cell section, I'm going to skip down a little bit and come back to this stuff. This is essentially the picture that shows activation of B cell. And let me enlarge this so we can see it a little bit better. There we go. So we have 3 different B cells here. And the naive B cells, once they've passed through the developmental processes in the bone marrow so that they don't recognize self-antigens, they look like this. The antibodies are on their cell surface and they act like receptors. So in the book here, they're calling them B cell receptors, but they're really just antibodies. In the picture here, they have one cell with blue colored antibodies, one cell with red colored antibodies, and one cell with purple colored antibodies. And the colors there to designate that these are different cells and therefore react with different antigens. And so we have all of these triangular shaped antigens all over the place, but they're only binding to the red cell, red colored antibodies, which means that they are not the antigen that the blue or the purple ones react with. When the naive B cell reacts with an antigen across its receptor antibody, it then will go into its activation. Now, this picture leaves out the role that helper T cells play, but helper T cells do help activate them by releasing that interleukin that I just mentioned, like IL-2 most likely. And so that leads them to start to proliferate or divide into multiple copies. These resulting copies are still making the red colored antibody because they only make that antibody. And then they end up making a small population of memory B cells and a larger population of the active B cells that once they're active and they've developed the secretory mechanisms to pump out a bunch of antibodies are called plasma cells because they secrete antibodies into the plasma. That's where the name comes from. While all of the other cells here are kind of these round looking things, the plasma cells are kind of oval and they have this squiggly thing in their cytoplasm. That squiggly thing is supposed to represent, not that you'd be able to see under the microscope, but it's supposed to represent the endoplasmic reticulum and Golgi apparatus, which is necessary for protein secretion. So they have to develop the ability to secrete those proteins in high numbers. That's what we see, and they start churning out a bunch of antibodies. Okay. After you've developed that immunity to whatever originally activated the cell, if you meet that antigen again, the memory B cells can then go through the process much more quickly, and you get a bunch of plasma cells that release more copies of the antibodies into the surrounding tissue. Okay. So earlier, when we were looking at the T cells, we saw activated T cells and memory T cells. Here we see the same thing with B cells. The activated B cells are called plasma cells, and the memory B cells are available to react to future infections of the same thing. Oh. Sorry, I closed this tab, and so it's going to mess things up a little bit for a second. Okay. So I skipped down here to show you where the B cells come from. Now I want to go back up here and talk a little bit more about the antibodies. And this table is showing you 5 different types of antibodies. Now, when we're talking about types of antibodies, we're talking about differences in the constant region part of the antibody. A single B cell is going to make its antibodies that are unique for what's in the variable region of the antibody, which is where the antigen binding sites are up here. What's different between these 5 different types of antibodies is really what's going on down here at the other end, and it's how they interact with each other. Now, I'm actually going to talk about them a little bit out of order from this table because I want to talk about them in order of their relative abundance. So the second column there, which is called IgG, is the most prevalent type of antibody that any cell makes, any B cell makes. Let me step back for a second. IgG. Ig, capital I, lowercase g, is an abbreviation for immunoglobulin, which is just another word for antibody. And then the 5 different classes are indicated by the letters after them. So IgG, IgM, IgA, IgE, and IgD. Why it's not IgA, IgB, IgC, IgD, IgE, I don't know. But there's IgA, IgD, and IgE. So maybe what was B and C got changed to M and G. But for whatever reason, this is how they're designated. Now, IgGs make up about 80% of the antibodies that are found in your system. They are usually monomers, which is what the picture at the top is showing you, a single antibody. 2 light chains, 2 heavy chains, Y-shaped formation, typical antibody. They are important because they can interact directly with phagocytes. So when they bind to their antigen, they will also make the phagocyte bind to them directly, which makes it easier for the phagocyte to phagocytose whatever they're attached to. The next type is IgM. IgAs, which makes up about 13% of what's in the body according to this table. I'm fine with these numbers. I'm just going to say according to this table because I've not double-checked the numbers. IgAs can be found in monomer form, but just as often they're found in a dimer form, which means 2 of them linked together in the center. Also, what's important is IgAs are usually found in secretion. So down there at the bottom, it says secreted into mucus, tears, saliva, and colostrum, which is referring to mother's milk. So in the mucus that's secreted in your mucous membranes, or saliva, which is obviously in your mouth, or tears, and you know where those come from, you're going to find these. And those are going to provide an immunity to things that you're exposed to at those barrier surfaces. And then IgAs are also passed through mother's milk. The majority of immunity that a newborn has is from actually IgGs that are passed across the placenta from the mother. But any new immunity that the mother develops after she's given birth can be passed on by IgAs through the mother's milk. The next most common is the IgM, which according to this table is about 6% of the antibodies out there. And what characterizes this is that 5 individual antibodies will all link together, forming a pentamer, or a 5-pieced macromolecule. Because 5 antibodies are all linked together, this pentamer will then have 10 antigen binding sites, 10 different spots where it can bind to antigens, possibly binding to 10 different antigens. Okay? So if you're infected with bacteria, you're not infected with just one bacteria. You're going to be infected with a population of bacteria. And so IgMs are great at linking a bunch of bacteria together so that phagocytes can destroy them all in one go. And if this IgM that we see in the picture here binds to 10 different bacteria, and then one of those bacteria is bound to another IgM, and that other IgM is bound to 9 other antigens, and then one of those, you can start to get a huge clump of cells all together, which makes it very easy for your immune system to get rid of them. Then at the end, the last 2 columns are the most infrequent. The very last column, IgD, at about 1% of the antibodies present, are found in monomer form, but most importantly, those are B cell receptors. So the naive B cell that I talked about a little bit ago that had an antibody on its cell surface, that was the type of antibody it had. Just the constant region part of the antibody is able to insert itself into the cell membrane, and it's a B cell receptor. And then the most rare form is the IgE, which makes up 2/1000th of a percent, I think that's what it says. Very, very rare. They are capable of interacting with mast cells and basophils. And IgEs are often associated with allergic reaction. Okay. So your allergic reaction is the inflammatory response. Okay. Overproduction of mucus, which is just a lot of fluid being produced by those types of cells, swelling, possibly redness, especially in your eyes or something like that. All of those things are really just inflammatory responses. The inflammatory response is made by histamine released by mast cells. So if you have IgEs that are sort of like receptors on the surface of mast cells, they can be directly activated by an antigen, which can be good if it's an antigen that's actually pathogenic. But if it's an antigen that's just allergenic, it's not something that would actually make you sick, and instead your allergy is because those mast cells are being activated by accident or being activated inappropriately. Now, if you look closely at what that says, it says antibody of allergy and anti-parasitic activity. Now, for what we heard in the podcast and what I sort of covered briefly a little bit ago, that was more about suppressor T cells, regulatory T cells and cytotoxic T cells. But here, this is an interesting thing. I don't know how it fits into the hookworm podcast information, but remember the whole point of what's going on with Jasper is that he has really bad allergies. And so he voluntarily gets a hookworm infection so that he doesn't have allergies anymore. And IgEs are tied to both allergy and parasitic infections. How that has anything to do with what's going on in the podcast doesn't, it's not related to the lullaby cells or attack cells necessarily, but rather this might be another mechanism that ties the two together. And so it's possible that that shows you some of what's going on. Actually, I want to take a second, something that I would have normally said after listening to the podcast, but in the last 5 minutes, let me just finish up there because it kind of goes off from this. I have been using that podcast for a long time. I think it's a good introduction to how complex immune function can be and how possibly having a low-grade parasitic infection might be helpful, that kind of thing. I've been using it for a while. 1 year, probably a couple of years ago now, I had a student come up to me after we listened to that in class, and she said, you know, I'm living proof that this is probably correct. Or she's not living proof, but she had direct experience with what the basis of that story was about. She grew up in Mexico City. When she was 19, she moved up to, I can't remember if it was actually up to Massachusetts or New York. Right? She came up to this area for college. when she was in college, again at about 19, one of the people in her dorm had really bad allergies. And she was convinced, my student was convinced for a little while, that this student with the really bad allergies and her friends who were feeling so bad for her having these really bad allergies were really just pulling this student's leg. She'd never met anybody with allergies before until she moved to the New England area and went to college. She grew up in Mexico City, not the most hygienic city in the world, I'm sure. Probably better than other parts of Mexico, but they say don't drink the water for a reason. Now, if you ever heard don't drink the water, and you've ever wondered, well, what do the people that live there do? They do, in fact, drink the water. Maybe not that much, but their immune system's adapted to that. Okay? So they might have a low-grade infection with some sort of parasitic worm that they got through the water. If you go down to Mexico and you drink the water, you're not immune to that, you're going to pay for it. But they've developed an immunity to it, and they can live with it. And maybe because of how their immune system is dealing with that less than hygienic environment, they don't have allergies. And this woman said she had never heard of somebody with an allergy, never met anybody that was experiencing an allergy until she came up here and met a child, a student who had been raised in America, probably with, parents that were giving them antibacterial soap left and right. So there is something to that might be true, just based on that story, which is not proof. But before you go, I just wanted to touch on one thing, which let me go into the presentation I have for the immune system, because that's the best place I have it. The book, and a lot of books that I've been using over the years, fail to do a good job of this. But when an antibody binds to an antigen, what happens next? And there's actually a few different things that can happen. And I have it written down in this presentation, which is why I'm pulling it up here. There we go. So when antibodies and antigens interact, that leads to a number of different things happening. And so this little list here, and if you put your stuff away and you're too lazy, you don't want to get it back out again, go to the Prezi presentations and you can pull this up there and write it down. But Plano is just the mnemonic I've used to describe this. Precipitation is when small antigens are bound up into large groups, and then phagocytes can clear them away. Lysis is what the classical complement pathway does, which I showed you earlier. Agglutination is when large antigens clump up, and then phagocytes can clear them away too. Precipitation and agglutination are basically the same thing, but one's for small things like proteins, the other's for big things like cells. Neutralization is when antibodies interfere with how the pathogen can do its job. So we're talking about a virus. If it's covered in antibodies, it can't latch onto the host cell and infect it. If we're talking about a bacterium, antibodies might interfere with its ability to move around or interact with its environment somehow. And then opsonization is actually a kind of very general term that's anything that increases phagocytic activity. I did not point it out in the book where it's mentioned, but in the innate immune system section, opsonization is there in bold case, and so it explains in the book there. But an antibody can interact directly with phagocytes to make it easier for phagocytes to engulf whatever they're attached to. Complement protein C3b, I described it briefly as being able to do the same thing. And it used to be, I introduced the term opsonization there, but those are the different things that can happen. Really, one way of thinking about it is any of the other 4 are going to lead to phagocytosis also. So we can sort of think that that is opsonization, increased phagocytosis. But sometimes that's all that happens. You have an antibody on something, phagocyte sees it and gobbles it up. Other times the antibody ends up being part of something else that helps get rid of the antibody. So this is just a quick run through. This information is in the book, but it's not organized quite this way. I like this way of organizing it because you can think of these major things all together. But all of that stuff is actually talked about in the book. It just spread out a little bit more in the text there. So I just wanted to finish with that. I'll leave this up on the screen if you want to write it down, but if you're, I'm done presenting things, you can head out for the day.